So today we're going to start talking about the T cell receptor. And the point of kind of talking about the T cell, about MHC, which we've been doing, is that we are really thinking about the antigen that is bound by the T cell receptor. And so that antigen might be MHC class 1 plus peptide that will be activating a CD8 T cell, or it might be MHC class 2 plus peptide that could be activating our CD4 T cell. And so really our goal here is to be getting to T cell responses and T cell actions. Again, you can see in this figure from your textbook that distinction between the CD8 T cell and the CD4 T cell, which we will be seeing more and more of. At this point, just you need to know there's the CD8s and the CD4s. T cells are able to recognize MHC plus peptide by using their T cell receptor. And you can see the T cell receptor in the middle here. The T cell receptor has two different chains. One of which we tend to call a heavy chain, one of which we tend to call a light chain. And that some of the details of that are a big part of what we're going to talk about today. You can see here that they are labeled in this image as alpha chain and beta chain, but they will not always be alpha chain and beta chain. We'll see some details on that later. But there are always two. One of them is always a heavy chain, one of them is always a light chain. They have a variable region and a constant region. They're connected by a disulfide bond. They have this really short transmembrane region and a really short cytoplasmic tail. And they have the three little complementarity determining or CDR loops at the ends that would make contact with antigen. And in a lot of ways, that T cell receptor is structurally very similar to an FAB portion of an antibody that also has a transmembrane domain. You can also see this in a ribbon diagram on the right. So you can see we've got the immunoglobulin domains of the constant regions, the alpha and the beta, the immunoglobulin domains of the variable region of alpha and beta. Coming together, we've got our three CDR loops that are projecting up to contact MHC plus peptide, which is our antigen, to make this process happen. And so we're thinking about this interaction between MHC and peptide, that complex, with the T cell receptor. As I mentioned to you previously, the T cell receptor has rather low affinity for MHC plus peptide. And so here you're seeing a sort of image trying to indicate different affinities of different biological molecules. And what you should notice is that T cell receptors are on the weak side, while other types of biological molecules bind much stronger. And in fact, the strongest binding molecules aren't even on here, and I don't know why that is, but that's okay. And as a result, if we want to actually have a T cell with its T cell receptor binding to an infected cell or an antigen presenting cell in the real actual body, the T cell receptor needs some help from some other proteins to get some additional strength to hold those cells together. And so we are going to see a lot of proteins that will work with the T cell receptor in some way. Some of those proteins are proteins that we're going to refer to as co-receptors. And the co-receptors, if I'm being official, the co-receptors are usually this CD4 that's shown in blue here, and all of these kind of pinkish proteins listed as CD3. It's down here, but you'll see those in exact detail in more slides, so don't get super worried about them right now. But co-receptors are really kind of helping the T cell receptor send its own primary signal. They're kind of helping send the primary main signal through the T cell receptor. There may also be other proteins that are kind of sending additional signals, like add-on signals. You can imagine this as, the icing on the cake kind of signals. They're known as co-stimulators. And so we will see the action of some co-stimulators as well as parts of the co-receptor. When we talk about any of these proteins, one thing that you're going to want to sort of pay close attention to is with each protein we think about, we're going to be like, oh, well, this protein binds this and this binds this. You want to remember which one's on the T cell and which one's on the antigen presenting cell, either the infected cell or the, professional APC like dendritic cell. And so whenever we think about protein-protein interactions, make sure you note which cell each of these proteins is on, because that's going to be really key. And you can even notice on like old exams and stuff, sometimes I'm like, put the proteins on the cells. And so to understand some details of this process, we need to think a little bit about the T cell receptor. Just like we did with the B cell receptor, we're going to think a little bit about a couple of details regarding signaling with the T cell receptor. Next week, we're going to have a whole day where we do even more details of signaling. But right now, we're just going to talk about a couple big picture issues. And we're also going to spend time talking about how the T cell receptor is made in the T cell. So if we look first at this image of the T cell receptor, you can see we've got our two chains, we've got our disulfide bonds, we've got our CDRs, and this is a good representation of this receptor. When we talked about the B cell receptor, I told you that the B cell receptor was awesome in terms of being a receptor because it's like so specific, and we went through all these fancy processes to make this ligand binding part of the receptor. But I also told you it had some ways that it kind of sucked as a receptor. And so what were the things that the B cell receptor in isolation like this was missing? Yeah, David. So one issue is that we didn't have much in the way of a cytoplasmic domain. And so it was really hard to kind of have signals going into the cell because we have this really tiny cytoplasmic domain. It's like 3 amino acids. It's not actually long enough to do anything. In particular, there's no enzymatic activity. There's no tyrosine to get phosphorylated. None of that. What else was missing from the B cell receptor? It makes sense that this one is less familiar to you because I spent less time thinking about it. So the other thing that I told you guys that was missing with the B cell receptor was actual enzymatic activity like a kinase. So there was no kinase. So there was no kinase and there was no intracellular domain to get phosphorylated. And if you remember, the B cell receptor solved the intracellular problem, the intracellular cytoplasmic tail problem, by traveling with two partner proteins, Ig alpha and Ig beta, that had cytoplasmic tails that could get phosphorylated. And I just said, and the kinase, trust me on that, because I didn't want to get into the details of the kinase. So as you look at this image of the T cell receptor, what do you notice about this image of the T cell receptor? Yeah, Jonah.
It seems to have pretty much the same problems.
Same problems. We have almost no cytoplasmic tail, so we've got no great way, nothing that's going to get modified to keep transducing a signal inside the cell, and we have no kinase. The way that the T cell receptor solves these two problems, just like with the B cell receptor, is that it travels with some partner proteins. One of the, I say one of, but that's a little bit of a not correct phrasing. One of these partners is something called CD3. CD3 is actually a complex of 6 proteins. There's one called delta, there's one called gamma, there's one called epsilon, and there's 2 copies of epsilon, and there's one called zeta, and there's 2 copies of zeta. And so you can see we've got delta epsilon, gamma epsilon, and zeta zeta here. Zeta barely has any extracellular domain. Delta epsilon and gamma do have a little bit of an extracellular domain, though we don't care much about them. We don't think about them as binding anything. Don't worry about their extracellular domains. The key thing about all of these proteins is that they have nice long cytoplasmic tails. So this figure shows them with some nice long cytoplasmic tails on delta, epsilon, and gamma, and you can see zeta being mostly cytoplasmic. This image also shows that. You can see our nice cytoplasmic tails on gamma, epsilon, epsilon delta, and zeta. And you can see that this picture shows them as having these yellow rectangles on their cytoplasmic tails. So delta, epsilon, and gamma each have one yellow rectangle as part of a cytoplasmic tail. You can see zeta has 3 yellow rectangles as part of a cytoplasmic tail. And those yellow rectangles are called ITAMs. ITAM, as an acronym, stands for I guess it's technically immuno. ITAM stands for immunotyrosine-based activation motif. Motif basically just means it's a part of a protein. That's not useful to us. Immuno, these are part of the immune cells. Again, not like really useful for us. Tyrosine-based, these all have the amino acid tyrosine in them, which is really good at getting phosphorylated. So these are all providing, you can imagine, these are all providing a tyrosine that is a place to get phosphorylated. So now we've got lots of cytoplasmic areas that can get phosphorylated. You can also notice that ITAM has an A for activation in it. Later on in the semester, we're going to see something that is called an ITIM, which is an immunotyrosine-based inhibition motif. So the point here is that if it's an ITAM, we're turning stuff on. We're turning on the T cell. Just for future reference, in case you haven't sort of thought of this before, tyrosine as an amino acid, A is a really good amino acid at getting phosphorylated, so that's why I'm going to mention it a bunch. It's also possible that I might sort of be drawing something out. The 1-letter abbreviation for tyrosine is Y. So if I start putting stuff with Y's on it, like random Y's, that's the tyrosine. One thing that's really cool about CD3, this is also true for Ig alpha and Ig beta with the B cell receptor, but it was first described for T cells and the T cell receptor, and it was actually one of my friends in grad school who described it. So in my mind, this is like the place where it's important, even though, yeah, it happens in B cell too. Is that even though this is actually 8 separate proteins, 2 of them, the alpha and beta chain, are covalently linked by a disulfide bond. This whole thing diffuses together in the membrane. The proteins don't like ever separate and get apart from each other in the membrane. They all stay diffusing together, even though they don't have covalent bonds other than between the actual T cell receptor. And turns out the way that works is that typically in a transmembrane domain, you don't have a lot of amino acids with charges, but these proteins all have amino acids with charges, and they attract each other, and so they actually stay together through those attractions. And so you can see that there are different charges in the transmembrane domains of each of these proteins based on certain amino acids that are there, and this all is held together. Even though there's non-covalent interactions, this will all diffuse together as one thing. And so even though the T cell receptor doesn't have its own cytoplasmic domain, it's always diffusing around with its friends who are bringing the cytoplasmic domain. We can also think about one other part of the co-receptor for the T cell receptor. And so what you can see is that the T cell receptor really couldn't send a signal without CD3. So that's kind of what co-receptor means, is it's part of sending that original signal. The other part of the co-receptor is either CD4 or CD8. And we've already seen CD4 and CD8 when we talked about MHC class 1 and MHC class 2. And we talked about these as these proteins that were stabilizing the interaction between the T cell receptor and MHC, which is correct. You can see CD8 is binding to class 1, CD4 is binding to class 2. In either case, the co-receptor protein is binding to a conserved domain at the back of the MHC molecule and sort of stabilizing this interaction next to the T cell receptor. Unlike the CD3 molecules, and again, notice that CD4 and CD8 are on the T cell, while MHC plus peptide is on the infected cell. Unlike CD3, CD4 and CD8 don't have those charged residues in their transmembrane domain, and they don't spend all their time next to T cell receptors on the surface of the cell. These are proteins that are not bound to each other in any way, and so they're going to diffuse like totally separately, randomly through the membrane, and not always be next to each other. But if, in the case of CD8, if MHC plus peptide comes around, T cell's going to bind to it, and CD8's going to bind to it. That's going to sort of force them to be next to each other, where they weren't next to each other before. They might have been like far apart from each other before. Now, they're both binding to the same thing, so they both get forced next to each other. Similarly, CD4 and T cell receptor were going around differently around the cell, diffusing not next to each other. And when MHC class 2 plus peptide is around, T cell receptor binds and CD4 binds. And this brings the two of them together. This is the induced proximity model of signaling. I know, and that's something that I think students don't always like click or get quite as early. And part of the reason is because it's really hard to draw a textbook of them like far apart from each other and then like moving close. If you're looking at like a static drawing somebody made, they're probably going to be kind of close because they have to be in the same drawing. And so this is a version from your textbook. This is trying to show them as far apart, and now they're close together. Yeah, it doesn't look that far, but they had to put them in one picture. And so what you can see is that originally the T cell receptor and CD4 are not totally directly next to each other, but when class 2 comes around, you can see that now T cell receptor and CD4 are pulled next to each other because they're both binding to class 2. So their extracellular domains are basically pulled next to each other. This would be the same if we were talking about CD8 and MHC class 1, but this image is showing us MHC class 2. Because the extracellular domains are getting pulled together, the intracellular domains get pulled together too. It's not like the extracellular domains get together and the intracellular ones stay apart. the extracellular part of CD4 and CD8 gets pulled close to the MHC and to the T cell receptor. And so the intracellular part gets pulled close to the T cell receptor. And so you can see not only is here's the intracellular domain being far apart, here's the intracellular domain being close together. That intracellular domain of CD4 or CD8 interacts very closely with a kinase. It's not the actual same protein. The kinase is just really a tight interaction. You can think of them as always traveling together. And if you want to know the biological reason, the only biological reason I got for you is because I say so. But just imagine that kinase is always sort of attached to either CD4 or CD8. And you can see that kinase here being called LCK. If I'm being super technical, if we were in a CD8 T cell, the kinase would not be LCK. It would be a kinase that is closely related to LCK. We're going to call them both LCK. But technically, the one that's on CD8 is a different kinase. But that's okay, we don't care. We just know that it's this kinase LCK. In this image, you can see our yellow rectangle areas of those ITAMs that are part of the CD3 molecule attached to T cell receptor. And when we pull CD4 in this case, or CD8 if it was a CD8 T cell, when we pull that co-receptor close to the T cell receptor because of the interaction with MHC plus peptide, that pulls this intracellular domain close. And now our kinase, LCK, is close to those ITAMs, is close to those tyrosines that were on the ITAMs. And this kinase can actually add phosphates to those tyrosines, which are depicted here as pink dots. So the kinase can phosphorylate the tyrosines, which you can see happening here. And that happened because this kinase that was attached to the co-receptor CD4 or CD8 was pulled into close proximity of the CD3 co-receptor. Phosphates have a different shape, which you can see here. See, there's no circly shape, but now there's a circly shape. They also have a different charge. And so when these tyrosines get phosphorylated, that provides sites for more signaling to happen. And so you can see there's a change in the cell and more stuff can happen. And we're going to talk in detail about what the more stuff is next week. But here, I just want you to see the importance of CD3, CD4, and CD8 in turning on the T cell and how they all interact with the T cell receptor and give it the intracellular domain and the kinase. Do you have a question, Juliana? Did I answer it?
Does the CD4 or CD8, does that have like a strong affinity towards the MHC?
It does, yeah.
That's why they're
Yeah, it does. It does. It has a very strong affinity to MHC. All right. So that's kind of one thing you need to know about the T cell receptor and how the T cell receptor works. But we also need to think a little bit about how the T cell receptor functions. So here you can see, again, just views of the T cell receptor. You can see our transmembrane domains, our short little intracellular domain, our disulfide bond, our two immunoglobulin domains, making a variable domain and a constant domain on each of the heavy and light chains. It just as I've been mentioning, we've got CDRs on both chains. So we have a CDR 1, 2, and 3 on the alpha chain because this is an alpha beta TCR, and a CDR 1, 2, and 3 of the beta chain because this is an alpha beta TCR, all up there to interact with MHC plus peptide. Here you're seeing in this green and red are the two different chains of or two different parts of our MHC molecule. Yellow is the peptide, and here you're actually seeing where the CDRs are binding, And you can notice that the CDRs are actually making interactions with both the MHC molecule and the peptide. So it is a binding that's happening to both of them. And we even know that the T cell receptor has a specific angle that it likes to interact with the MHC molecule at. And so this is sort of the angle, this box is what the T cell receptor would be seeing across the MHC plus peptide. And so you can again see that it is binding sort of across all of MHC plus peptide. It's not just like binding this loop, this corner over here. It's binding across the whole thing, including the peptide binding cleft. You can see that's true for both class 1 and class 2. I will mention when we talk about autoimmunity later on that there are some really well-known cases in autoimmunity where part of the problem is that the T cell and the antigen presenting cell are binding at the wrong angle, and so the angle is actually an issue in a few cases. When immunologists first started thinking about this, they started to do some math problems and come up with the same kind of math problem that they came up with the antibodies in terms of how many T cell receptor genes there were versus how many genes there were in the genome. And they were like, oh, crap. And they were really worried for a while that they were going to eventually discover another whole process that makes diversity in T cell receptors, and they were going to have to learn another whole process that makes diversity in T cell receptors, and they were really sad about that possibility. But they eventually found out that the T cell receptor was made by VDJ recombination, just like happens in B cells and antibodies. And they're like, oh, sweet, we already learned that one. We don't have to learn more. And so in a lot of ways, the details of VDJ recombination are very similar for the T cell receptor and for the B cell receptor. So I'm going to show you both similarities and differences. I will also tell you that when I was in grad school TAing this class, we would always throw like a really hard VDJ recombination question on exam 2 because we were like, haha, it's still relevant because it was happening in T cells. Probably not going to do that. But note that really all the things you learned about VDJ recombination in B cells are technically all still sort of relevant as a part of what we're seeing in T cells. So we've got two different chains being made. One of them has a V, a D, and a J. One of them has a V and a J. So even though they're basically the same size or very similar in size, we tend to call the one that has the V, the D, and the J the heavy chain, and the one that has the V and the J the light chain. And in a lot of ways, they are quite similar to what we see with the antibody heavy and light chains. Similarly, if we think about how the CDR3s are encoded, CDR1 and CDR2 from beta are just based on which variable region or V region is chosen, and CDR3 in the beta chain is based on which V, D, and J have come together. Similarly for the alpha chain, CDRs 1 and 2 are just based on which variable region or V region we're using, while CDR3 is made at the junction of the V and the J region. So again, same exact thing that we saw with the B cell. And one other thing that I will point out here is that in an alpha beta T cell, in a T cell that has alpha and beta chains as part of its T cell receptor, and alpha beta T cells are the majority of T cells, or at least they're usually, if you've ever heard anything about T cells in your life, you were hearing about alpha beta T cells. In that case, beta is the heavy chain and has the V, D, and J, while alpha is the light chain and has the V and J. And you can also see that here as well. So here you are looking at the mouse beta chain and the mouse alpha chain. Pretty similar to what you've seen in antibodies. It is a little weird that there has been this duplication in mouse. I'm honestly never going to really talk about that. But yes, technically the Ds and Js and constant regions are duplicated or in multiple places. But just to imagine it looking like the heavy chain in T cells or in B cells. I'm not going to like care that you know about this duplication. And we've got our Vs and our Js for the alpha chain shown here. So in that way, everything looks really similar to what you've seen before in B cells. Sadly for all of us, there are some things that are unique in T cells. And so this is the first thing that's unique in T cells. So what I'm showing you here is basically the same thing that I showed you on the previous slide, the mini gene segments that we've been thinking about with their corresponding RSSs, because since VDJ, it's the same old details of VDJ recombination, RAG1 and RAG2 are still involved. We still need RSSs. You can see on our alphas that we have Vs and Js because it's a light chain, and the V alphas have 23 base pair RSSs. The J alphas have 12 base pair RSSs. All good. But you might notice something fishy when you look at the beta chains. So what do you notice when you look at the beta chains? How is what's shown here for TCR beta different than what you saw for the immunoglobulin heavy chain? Yep, David.
For the B cells, the D portion had the same, so one of those 12 or 23 was the same on both sides.
Mm-hmm.
That's different, and the B and J section is not like that.
Yeah, so in B cells, the RSSs looked like this, where the D segment had the same RSS on either side. V and J had the same ones, and so you would, put your D with your J, then you'd put your V with your D, and you'd all live happily ever after. I'm going to redraw this drawing to put in one other thing, and then I'm going to ask you a question about it. Okay, so here is my updated B cell heavy chain gene locus. Because of the way that the RSSs are set up in B cells, there are two things that are not possible. What are the two? There are two kind of weird things you could imagine happening that are not actually physically possible for the B cell. What are those two things? Yep, Shelby. Okay, so one problem is that if we do the rearrangement, you can't choose a different D segment. And that actually is going to be true in T cells too. Okay, so that one's going to stay true in T cells because you're going to delete the intervening sequence. So yes, that we're going to see again. Yeah, Jonah.
The B and J can't be spliced together.
So what you notice here is that you can't put V and J together and skip D. That's kind of what Shelby was also getting at. Is you can't do V and J and skip D because the V and J have RSSs that can't go together. They have the same kind, and you need to have different ones to put it together. So you gotta have a D. You can't skip D. So that's one thing that's not allowed when we look at this B cell. There's a second thing that's not allowed. What else? And I drew it like this very purposefully for here. So what's the second thing that's not allowed? Yeah, Josh. Yeah, you also can't put a D with another D. You can't fuse two Ds because the Ds don't have RSSs that go together. So now if we think about what we would see for a T cell heavy chain, which we'll say is beta in this case, here's what it looks like. I think I drew that correctly. Yeah, I did. So you might now, as you look at this in the T cell, have two questions for me about what happens in VDJ recombination in T cells. So what questions might you have for me about what might happen in VDJ recombination in T cells? Yeah, David.
How does it avoid putting two Ds together?
So one question is, well, what's the deal with Ds? Do we have two Ds put together? David assumed that we don't, but the question is actually, do we have two Ds put together? And the answer is actually yes, we do put D. So T cells have the additional ability to get even more diversity by going VDDJ. So they will have what's known as DD fusion sometimes. So DD fusion is actually an option in T cells. And so that's one thing that's unique that we see in T cell heavy chain recombination that we don't see in B cell heavy chain recombination. So what's the other question that you might have for me? Yeah, Jonah. Can the beta chain just skip D? Can we have a V with a J and just skip D altogether? We have never observed it. And so we think the answer is no, it is not possible. I could give you some molecular genetics explanations of why I think it's not possible, of why we think it isn't possible, but we don't actually officially know the answer. But the answer is it doesn't happen. Why it doesn't happen, how we keep it from happening, is an interesting question, because if you look at this, it should be a choice, but it doesn't happen. However, DD fusion does happen, and we do see DD fusions occurring. Otherwise, when we think about T cell diversity, we really are getting a lot of the same types of things going on that we saw with B cells. The exact numbers of V's, D's, and J's is different. The exact details of how junctional diversity works is different. So we get a different number, but it's the same general idea of using combinatorial diversity and junctional diversity to get this diversity. And we can sort of compare and contrast what we saw with B cells and T cells. Do we have multiple V, D, and J's that we're putting together in different combos? Yes in B cells, yes in T cells. Do we have a light chain? Yup. Do we have a heavy chain? Yup. Do we need RAG1 and RAG2? Yup. Do we have P and N nucleotides? Yup. But weirdly enough, in T cells, we can get multiple Ds put together, so we can get these DD fusions. We're going to have to talk a little bit about allelic exclusion because the T cell receptor, particularly TCR alpha, is not great at allelic exclusion. But then the other thing that is different is we never actually secrete the T cell receptor. So like with B cells, remember how there was B cell receptor or antibody? that came from the same gene, we don't have a secreted version. We just have the T cell receptor that's on the surface. So yes, we secrete the product of the B cell receptor. No, we don't with T cells. And thus, since we don't have a secreted product in T cells, it can't do different things based on its constant region, like IgM versus IgG. And the B cells undergo somatic hypermutation and class switch. Those things we talked about later in the life of a B cell that we haven't really gotten into any details of, but I said that happens later. They happen in B cells. They don't happen in T cells. So once the T cell has finished this process in the thymus, it is done. So in that sense, things are pretty straightforward in thinking about what's going on with T cells. And as I've already told you, there are a lot of different types of T cells. I've introduced you to our two major types of T cells, the CD8 T cell and the CD4 T cell before. And I've told you briefly that there's even more types of T cells that exist that I haven't sort of gone into yet. One of those other types of T cells I have to tell you about is a type of T cell that uses a different kind of T cell receptor. So kind of your classic T cells that most everybody thinks about, particularly when they're thinking about CD4 and CD8 T cells, are T cells that are known as alpha beta T cells. And alpha beta T cells have a T cell receptor with two chains, a heavy chain and a light chain. The chains are named alpha and beta in an alpha beta T cell. Alpha is the light chain, beta is the heavy chain. But there are some other T cells that use a different receptor. They use some different receptor genes. And these receptor genes are known as gamma and delta. And so we also have some T cells that are known as gamma delta T cells that use gamma and delta chains to make their T cell receptor. Again, one is a light chain, one is a heavy chain. In this case, delta is the heavy chain and gamma is the light chain. And so sort of classic T cells that most everybody thinks about are alpha beta T cells, but there is this other unique group called gamma delta T cells. I'm not going to say, like, for the whole semester, there's not a ton that I mention about gamma deltas. We're mostly talking about alpha betas. But how the T cells do VDJ recombination when they actually have 4 different chains they could use becomes an interesting question for the T cell. And so this is really another way of kind of showing you something I said on the previous slide, which is you can see the information about the alpha, beta, gamma, and delta gene segments. You can see that beta and delta are both heavy chains because they have D segments. Alpha and gamma are not heavy chains. They don't have D segments. The weird RSS issues that I told you about earlier with the alpha and betas are also true for gamma and delta. So all of that is the same. But there's one other fun little quirk to all of this. I'm going to show you a table on the next slide that involves some information from both humans and mice. And I'm going to ask you a question about those data. When I ask you the question, I want you to know one specific thing, however. I want you to think about this in terms of, I want something that's true in both humans and mice. There's something else that students always like that jumps out to students. And it is true. But the key thing that we need to notice in this table is something that is true for both humans and for mice. Okay? And this table is showing us the chromosomal locations of these 4 gene regions. And so if you remember, when we thought about B cells, I don't remember off the top of my head. I think, what was it? Heavy chain was on like chromosome, I don't know, was on some chromosome number, and light chain was on some other one, right? So here I'm telling you what chromosome number. Alpha, beta, gamma, and delta. And so these are 4 different sets of V's, 4 different sets of J's. In the case of beta and delta, 2 different sets of D's. So we got lots of V's, lots of D's, lots of J's. This is where they are on the chromosome. There's something unique about some of these gene segments or some of these loci. What do you notice that is true in both human and mouse with these loci? Yeah, Shelby. What do you mean by the light chains are in the same spot? So beta and delta are heavy chains. Delta is a heavy, not a light. You're pretty much, but rephrase what you just said. Mm-hmm. Alpha and delta are on the same chromosome, right? Yeah, it just so happened, yes, those are the same, but we don't actually care about that. But we do care about the fact that alpha and delta are on the same chromosome. They're on the same piece of DNA. And it's more than just that alpha and delta are on the same chromosome. If we look at the gamma and the parts of the chromosome that have all of these genes, we really only ever look at 3 locations in the genome. So we can see here is beta. We got our V betas, we got D beta, we got J beta. Here's gamma, we got V gamma, J gamma. But then at the top, here we've got V alphas, and then we've got V deltas, and then we've got D delta, and then we've got J delta and C delta, and then we've got J alpha and C alpha. Delta, the whole delta locus, is in the middle of the alpha locus. And this is true in all of these different organisms. So let's imagine for a second that we have a developing T cell that does VJ rearrangement, because it's a light chain, for alpha. Let's imagine that we are going to join together V alpha 2 and J alpha 2 to make an alpha chain. When we do that, what happens with delta? It's cut out. So if you try to do VJ rearrangement of the alpha chain, you throw away delta. You cut out delta. You can see that here as well. That the entire delta locus is in between the V's and the J's of alpha. And so if a cell does rearrangement of its alpha chain, it actually deletes and throws out the DNA for the delta chain. This is from an older version of your textbook. This is from the current version, where you can see all of the intervening DNA that includes all of delta gets deleted. What that means is during T cell development, our T cells have to make a choice. They have to make a choice. Are they going to be an alpha beta T cell? Or are they going to be a gamma delta T cell? Because you can't make both alpha and delta. You can either make alpha or delta. They're using the same region of the chromosome, you gotta make a choice. Am I going to use alpha or am I going to use delta? Because I can't use both. And after the cell makes that, and so how the cell makes the choice of am I going to do alpha beta or am I going to do gamma delta is a big part, is a separate thing that we're going to have to see in the process of T cell development. If the cell chooses to be an alpha beta T cell, it will do all the T cell things that we'll talk about for much of the semester. It will do these T cell things. If the T cell decides to become a gamma delta T cell, it will become a gamma delta T cell. And gamma delta T cells seem to be really unique. They tend to be found mostly only in barrier tissues. So like you have a lot of gamma delta T cells in your skin and in your GI tract, and they have a lot of almost innate-like functions in terms of potentially being a little bit less specific, but you can also see they might recruit neutrophils, they might do some innate immune responses. They they develop pretty early in development, and so they may be really key in young organisms. There's a lot that's not super well known about them, partially because, as I mentioned, they tend to be in barrier tissues, not as much in the blood and in other types of tissues. And so if I wanted to study gamma delta T cells from our mouse, that remember when we got our mouse and we got the spleen, we can get lots of alpha beta T cells really easily from the spleen, but like what we might have had to do to get a lot of gamma delta T cells is like skin the mouse and grind up the skin. Does not sound very fun, right? You can imagine it can be really hard to pull these cells out of the tissue, and there's a lot that we don't know about them because we haven't always pulled them out of the tissue. Similarly, think for a second about how in the world you get them out of humans. Whether you are trying to actually pull them out of the walls of people's GI tracts or pull them out of people's skin. They can be harder to isolate, and so they can be much harder to study. And so they are sort of a little bit unique. They'll come up again in one other lecture, other than sort of when we think about aspects of T cell development. So we're, just like we did with B cells, we also have to think about development and selection of our cells. Officially, Friday is T cell development day, but there's a lot to be said about T cell development. So I want to talk about a few at least big picture pieces of T cell development now before we go into sort of the steps. Remember the pro B cells and pre B cells and who does what when. That's going to be more next time. But there are some big picture things we need to think about with T cell development now. And one of those things is here, which is that T cell development and selection, so our central tolerance induction as well as all of the developmental stages, happen in the thymus, not in the bone marrow. So if you remember, the stem cells started out in the bone marrow and became B cells and went all the way through B cell development in the bone marrow. If, however, one of those lymphoid or one of those progenitor cells, one of those stem cells, gets a signal that says you should grow up and become a T cell, the cell leaves the bone marrow and does all of the steps of T cell development in the thymus. You saw the thymus in the mouse. The thymus is this bilobed organ, so it has two little lobes that sits on top of the heart. You can also see it in this image here. You can see the thymus having its two little lobes and sitting on top of the heart right here. Sometimes people get confused with the thymus and the thyroid, which is an endocrine gland. The thyroid is up here in your neck. It is not the thymus. So this image does show the two of them. And so all of the steps that we're going to be seeing for T cell development are going to be important steps that are happening in the thymus. Sometimes people like to talk about the thymus as like T cells have to go to college to get their education and do their development. as we will see when we talk more about T cell development next time, a lot of T cells, a huge number of T cells die in the process of T cell development and are not successful. And there is this book that's actually really funny about the immune system that's like for the lay audience, and that book calls the Thymus Murder University, because of how many of those developing T cells die. There are lots of things to say about the thymus. So here you can kind of see our, we have our original hematopoietic stem cell. It's in the bone marrow. It could become a myeloid cell. It could just say, no, I'm going to become a lymphocyte. That cell could stay in the bone marrow and become a B cell, or it could leave and go to the thymus and develop into one of our many kinds of T cells. And so, that cell is traveling to the thymus. And when we think about our steps of T cell development in the thymus, we're going to be in the thymus a lot. You can see the structure of the thymus here. What you can notice is that the thymus is divided into two really big areas. One of those areas is called the medulla, and one of those areas is called the cortex. And they are easily visible as different under a microscope. I love this figure at the bottom. So when I was an undergrad, I actually worked in a lab that did a bunch of thymus stuff. So I felt like I, knew all the things about the thymus. And I'd seen lots of images like this one at the top, where I would see areas of cortex up here that I can't totally reach, and areas of medulla down here. And for some reason, I always thought that the thymus looked like a target symbol. And that like there was one layer on the outside and one layer on the inside. And I really like this image down here because it shows like there's medulla areas and cortex areas. It's not like one big medulla and one big cortex. It's not like the Target logo. You got some of each. And your textbook also has this nice image that's a cartoon version of it at the top. So if you look at the cortex and the medulla in either of our two microscopy images, what would you say is the big difference between them? Again, imagine for a second that you're my best friend's 6-year-old when you answer this question. Don't overthink this question. What is the difference between the cortex and the medulla? Yeah, David.
Color.
The color, right? The cortex in both of these images is a darker color than the medulla, right? That's actually kind of, if you can remember that, that will actually help you a lot. The reason why the cortex is so much darker is because the cortex has a higher density of cells. It's really crowded with cells. There's not much room for light to go through because it's so crowded. While the medulla has a less or a smaller density of cells. And particularly when I say cells, I mean the developing T cells. And so you can even see this in this cartoon up here. The developing T cells are in blue, and you can see in this cortex area, there's way more. The cells are way denser than they are here in the medulla. They're way more crowded in the cortex than they are in the medulla. And that's actually going to end up relating to what kinds of processes are happening in the cortex and in the medulla. That will be related to, or relate back to how many cells end up being in both of those places. What I also want you to notice about this image is that our developing T cells are making a lot of contacts with other cells in the thymus. There's all these cells that are shown as yellow and orange and red, even some of these pink cells. Those are known as the thymic stromal cells. or the stroma. We can also just say they're the structural cells of the thymus. And the developing T cells make a lot of contacts with the thymic stroma, the stromal cells. That is going to be really critical in what's going on in T cell development. But I want to leave you with one other interesting fact about the thymus. Though I do sort of snicker at this slide a little bit, which is this slide. So one other thing that's really interesting about the thymus is that the thymus changes with age. So when we were doing our mouse experiment, I had to be very specific in terms of how old our mice were for that experiment because I wanted you to look at the thymus. Because over time, the thymus changes. So this is a young thymus that you see in the middle, and this is an old thymus. The thymus actually gets basically invaded by fat cells, and you have less surface area of all of that thymus structure. This process is known as thymic involution. And the idea is that as you age, you actually make fewer T cells. You do less T cell development over time. In B cells, you keep doing the same amount of B cell development your whole life, but your thymic output, the amount of new T cells that you make, decreases throughout your life. And so this shows you kind of how much T cell production is coming out of the thymus at different ages. You can see, in the first year of life, the thymus is really pumping out T cells, decreases a little bit, then sort of 11 to 25, it decreases a bunch, and it keeps decreasing more. Many physicians were actually taught that the thymus stops working when you're about 10. That's actually, most doctors think that your thymus does nothing in adults. Part of the reason why I snicker at this is that when I was an undergrad, I worked in a lab that was studying the thymus. One of the things they actually did was for the entire 4 years I worked for them, they took blood from me every Friday. And so they could actually try to measure, like, in great detail in one person over 4 years what was happening. And so I was definitely making T cells at 18. And I definitely was even making T cells at 22. It just wasn't as high as before. And so if we had used mice that were too old for our experiment, the thymus, basically you wouldn't have been able to pick up with tweezers because it would have been so fatty, and you might not have been able to see it. It would have just fallen apart when you touched it. Like I said, there's this, your thymus definitely does decrease output as you age. I think that sometimes it's a little overstated. It's not like your thymus just stops 100% when you're 10 or something like that. One thing that's actually also really interesting about this is that when little kids have heart surgery, so if you have a small child who has open heart surgery, usually the thymus is in the way, and the surgeons just cut it out because it's in the way and it's not useful, which is a little bit makes me a little freaked out and angry. But that's actually why the lab I worked in had so many children's thymuses to study, because we got all of the thymuses that were being taken out of kids who were getting heart operations. We convinced the surgeon to give it to us instead of throw it away. Know that thymic function totally decreases with age. It is very hard to work with the thymus from an older individual, or particularly an older mouse. It's really hard. But sometimes I think it's overstated. And so if any of you guys end up, in med school, please don't think that it just stops completely in older adults, because it doesn't stop completely. Though yes, it does slow down. And we will talk about what events happen in that thymus on Friday. And please make sure you have a laptop with Excel for lab this week.